Welcome everyone. Gives me immense pleasure to welcome you all in this session on AMR and One Health. We'll talk about stakes involved and the way forward. I'm Yogesh Lakhotia, one of the co-founders of India Biotech Leaders of Tomorrow. I'm also a product manager at Elucidata. Very excited to see you all here. I think people are coming in slowly, and in the meantime, we'll get started and kickstart the event. Brief about IBLoT to start with. IBLoT is a non-profit initiative aiming to foster a community of biotech leaders of today and tomorrow to accelerate the growth of Indian bioeconomy. Through our initiatives, we hope to create a holistic understanding of bio-entrepreneurship, challenges and opportunities in the Indian biotech ecosystem. We're building a diverse community of Indian bioenthusiasts spanning entrepreneurs, scientists, academicians, researchers, investors, etc., so that they collaborate themselves more seamlessly. Right now, to start the event, I would definitely like to thank the sponsors. We are honored to partner Techinvention Lifecare Pvt and BioGrid Technologies. Techinvention is a young startup biotech company based out of India. It is one of the few companies which uses quite holistic approach for infectious disease management. Their focus has been on developing diagnostics, prophylactics, and therapeutics for infectious disease for developing countries. The company has numerous success stories and received multiple accolades of national and international repute. BioGrid Technologies is an upcoming biotech startup incubated in Toronto Business Development Center, Ontario. They develop innovative biosolutions with focus on enabling One Health. The team is dedicated towards discovery and development of diagnostics, prophylactics, and therapeutics against infectious disease in regions of America. So to give you a brief of the flow of event today, we'll have an introductory note from Mr. Syed. Following by that, we'll have Dr. Herman Barkema, who will speak on the importance of One Health with the focus on animal health and zoonosis. This will be followed by Dr. Urmi Vajpayee, who will shed light on AMR situation in India and globally, the efforts which she has taken towards AMR menace and some of the research work which she has been doing. Followed by these sessions, we'll have a panel discussions with our esteemed speakers, which will be moderated by Mr. Syed Ahmed. So without any further ado, I'll start introducing our esteemed guests today, starting with Dr. Herman Barkema. Dr. Barkema is currently a professor in epidemiology of infectious diseases at University of Calgary, with a joint appointment in the Department of Community Health Sciences of Cumming School of Medicine. He is also a guest professor at Ghent University and foreign expert at China Agricultural University in Beijing. Dr. Barkema's research focuses on prevention and control of disease in cattle herds, including AMR. He has published over 300 scientific manuscripts and lectured all over the world. He leads the University of Calgary Biostatistics Center, the Environment Research Team in Canadian Bovine Mastitis Research Network, the Alberta Jaundice Disease Initiative, and the AMR One Health Consortium at University of Calgary's. Right. Now moving to Dr. Urmi Vajpayee. She is currently the professor at Department of Biomedical Science at Acharya Narendra Dev College, University of Delhi. She is also the co-founder of International Bacteriophage Research Consortium. Her research interests include studying the potential of Mycobacterium tuberculosis, MER pathway enzymes as potential targets for developing drugs against pathogen, exploring antimicrobial potential for Mycobacteriophage derived endolysins. She has been the PI and several research projects funded by DBT, Delhi University, and has published a number of publications in reputed peer-reviewed journals. She has received a number of awards in her professional journey and has been routinely invited in several national and international platforms. Now moving forward to Mr. Syed Ahmed. He is the founder, director, and CEO of Techinvention Lifecare Pvt. driven by the imperative to drive positive change. Syed is fiercely passionate about using innovative approaches to deliver biotech products to those who need the most, especially in the developing world. He believes that novel phyolactics and therapeutics should not be a privilege and should be affordable for everyone. He founded Techinvention fueled by the desire to bring his mission to life. With equally passionate colleagues, he works to build novel pharmaceuticals capacity and infrastructure in the developing world. Syed has spent most of his career of over 23 years helping pharma companies with the capacity and infrastructure building. And as one of the foremost experts in the biotech consulting, he has spearheaded projects over 30 countries across Asia, Africa, and Latin America. Now, with this, let's get the session started. Let's keep it interactive. For any questions you may have, please put it in the chat box. And now I welcome Mr. Syed Ahmed to get the event started. Yeah.
Thanks a lot, Yogesh. I don't think there would have been a better opportunity to host this webinar. Today being the first day of the AMR Awareness Week, being November 18th. It's a Friday morning for Professor Barkema and a Friday late evening for us in India. And we have folks joining from Europe, Africa, who are somewhere in between. So it's a good afternoon to all of them or an early good evening to them. Of course, we have more than 75 participants already. So I wouldn't like to take much time. I'm sure all of them are here to listen to Professor Barkema and Professor Urmi. But rather, I would just take a minute to introduce our partners for the event. Our partners for the event are EBPMN and IBRC. EBPMN is Emerging Biopharma Manufacturers Network. This was constituted in the year 2019 when a team of manufacturers from emerging countries comprising of Butantan from Brazil, CNBG from China, Biovac from South Africa, all came together at the WHO headquarters in Geneva, along with UCAB. And I'm sure all of us are aware of UCAB, which is Utrecht Center for Affordable Biotherapeutics. They came together with the mission of enabling capacity building for biologics, which encompasses biosimilars and novel biologics in low-middle income countries. The organization has grown from strength to strength over the last 3 to 4 years. Today, we have more members, webinars, interactive sessions, and so on. We are happy to have EBPMN partner with us. The second organization, of course, is IBRC, International Bacteriophage Research Consortium. We have Professor Urmi, who is the co-founder. This was constituted in the year 2018, largely with the efforts of ANDC, which is part of the Delhi University. Incidentally, ANDC is also celebrating its centenary celebrations this year, 100 years. Wow, what an achievement, Professor Urmi, for the institution. And we also, they partnered with an organization called OHSL in Maryland, US, which is Open Health Systems Lab, with the objective of bringing novel concepts got to do with phages and lysines in the form of biotherapeutics, especially given the context of a depleting antibacterial pipeline, which is seen as the need of the hour, given the decade and the challenges that we are in. I would also take this opportunity to thank Innovation Council in Geneva. Jennifer Brandt and her team have done a fantastic job of promoting the event, because of which, although being a Friday, we have 80 participants across countries, across borders. So with that, I would like to leave the floor to you, Professor Barkema. Over to you.
Thank you very much, Syed. So it's a pleasure to be here, or to be here in a Zoom environment. I've never been in India, and I would love to visit India soon, and I will actually in the next year. So, and I'm really happy to do this in together with Professor Urmi, and yeah, I think we have very complementary expertise. So, thank you for that. Good. I'll start with sharing my my screen. Good. So, can everybody see this?
Yes.
Good. Then I'll see where. Yeah. Good. So my presentation is on antimicrobial stewardship and why One Health is so important. The COVID-19 pandemic that we all experienced was what we predicted that it would happen, that it would be a respiratory pathogen that would come. We thought it would be influenza, but that was not the case. So, you know, and that it would be a One Health event coming from animals. And these kind of surprises will happen. I put here some pictures of Canada, but we can do it from all over the world. The left top picture could also have been of Pakistan, for instance, with the flooding there. And we see forest fires and all kind of things happen in the world. So, and these events all come together and find an origin in things like climate change, but not only climate change, also all the travel that we do, all the animals that we send all over the world. So there will be a long-term impact of these events. So what we see here, what are we doing? You sometimes wonder. All this pollution in the ocean, and then because of deforestation, you get mosquitoes, and that cause, like here, Zika virus, particularly in Latin America, Ebola in Africa. And we have here avian influenza that is really happening all over the world, and of course, antimicrobial resistance. So a lot of things happen. So antimicrobial resistance, you know, we are predicting all the time that it will become a problem. So we really, really need to do something at it together. But it is one wicked problem. This is what we call here the confusogram. It is all the interactions that we have with humans, the environment, that determine how the transmission occurs of antimicrobial resistance from animals to humans, humans to animals through the environment. So, because of all these interactions between animals, humans, and the environment, finally, FAO, OIE, now the World Organization for Animal Health, WHO, and the UNEP decided that we need to work together to really get rid of the silos that we have in working together. So in the statement, also the G7 countries, they decided that we need to improve integration by strengthening a One Health approach across all aspects of pandemic prevention, and antimicrobial resistance is a pandemic, and preparedness, and recognizing the critical links between human and animal health and the environment. So what is One Health? One Health is an approach, a transdisciplinary approach to address problems at the intersection of people, animals, and their environments. And in veterinary medicine, we often realize that that's the case. We see it. But in human medicine, too many people in human medicine still think that people are shielded from those things, the bugs that occur in animals, but they start to realize that now. So the goal is to acknowledge the interconnectedness between those three pillars that we have, and the complexity of these connections, and really look at problem solving outside of the traditional siloed approach. If you look at, for instance, universities, we are all in silos. Engineering students don't know the veterinary students or the medical students, and often The human medicine students don't know the veterinary students and the other way around. So, and it is so important that they know each other, because to work together, you need to develop trust. And if you don't know each other, it's very difficult to develop trust. So you need transdisciplinary understanding and the development of practical solutions. We don't need jack of all trades, masters of none. We need people that are good in their area, but also learn how they can work together to first identify what really the problem is, and then together work towards solutions. So back to antimicrobial resistance, it's an increasingly pervasive issue worldwide. Antibiotics, of course, are one of the greatest medical advances of the 20th century, but we now see that we get these superbugs, we get a lot of resistance in livestock, in bugs, in humans with bugs, but also in wildlife. So these resistant bacteria, they don't stop when they are in livestock, they go to humans as well. And the other way around. So the same antibiotics are used in humans, animals, and the environment. And these residues of the antibiotics end up in the environment. We don't have to be that afraid that people get resistant bacteria in their body because of drinking milk or eating meat, if it's properly heated. But these bacteria are also end up in the wastewater through the manure and the urine of the animals. And most often those bacteria are not limited to one species. Think of Staphylococcus aureus, but we can come with a lot of those bacteria. So, and there are very many factors that contribute to the selection pressure of these bacteria. So we need to work together to really curtail the spread of antimicrobial resistance and look at the environment as a reservoir for resistance. And it's really important that we not only look at the kind of rich people that we typically are, because if you have a computer and you watch this presentation, you typically are not poor. But it occurs particularly also in people that live under poor circumstances. And it's not only because we think of those people, but it's also the antimicrobial resistance bugs that develop in these people will also go to the rich people. And also bugs don't stop at borders with the travel that we do all over the world. So we need to work together all over the world. So what has led to this development and antimicrobial resistance? Of course, the increased use. Often prescriptions were not taken correctly and stopped when most of the symptoms were gone, but the bacterial infection is still there. We see a lot in a lot of countries, and I lived in Zimbabwe and I lived in Costa Rica and a lot of other, and I visited a lot of other countries, and you could just get antibiotics there at the street corner. And so that is something we need to work on. Then there's a lot of prophylactic, preventative use, particularly in livestock, that needs to stop. However, that can only stop when the management of these livestock is good, because otherwise they still need this umbrella of antibiotics for the infections that occur because the animals are not in the best shape. Then we see a lot of antibiotics used for viral infection. We published also a paper on AMR in COVID-19 patients, and it was clear that because of the aspecific symptoms in the beginning of the infection, that antibiotics were used in COVID-19 patients, and antimicrobial use led to antimicrobial resistance. We see a lot of spread of resistant microbes in hospitals and farms. Hospitals are in a way the worst place to visit because if you want to get an infection, the sick people are there. But we also see it in farms. So, and then we have the residues of the antimicrobials that are in the environment, and if they are end up in drinking water, then people get low doses of antimicrobials, and that's ideal for development of antimicrobial resistance. So they are not the cure-all, they are the ones that we need to use as precision medicine in the case that they really work. So if you look at the picture of livestock, humans, and pets, we now know that antimicrobial use also in livestock leads to antimicrobial resistance, and also the same in humans and the same in pets. That's quite clear. We had some questions about livestock to humans, but that is also clear. And what we also see that if you stop using antibiotics in livestock, that we see antimicrobial resistance levels also genetically in those bugs go down, and also the levels go down in the people that work with these animals, but also of the people that live in the vicinity of these animals. So we don't know a lot about pets yet, and that's unfortunate because pets live so close to humans. But we know that also using pets makes antimicrobial resistance and can cause antimicrobial resistance in humans, but there are just a few reports on that. We don't know a lot about wildlife. We hardly ever measure in the environment, and particularly we don't know a lot about aquaculture. And think of fish being kept in cages in the ocean and where antibiotics are used. And what is the effect there on wildlife? So we have a lot of stakeholders. We all need to work together towards that. It's not just physicians and veterinarians and people working in the environment, but it's also other groups that need to work together. Pharmacists are, of course, an obvious one, but the pharmaceutical companies need to show their goodwill, and they do in general. Towards antimicrobial stewardship, because they realize that if this misuse in some countries continues to happen, that we get more and more antimicrobial resistance all over the world. So I couldn't find good data in India, but just to make the point, in livestock, even if you correct for the weight of animals, because a cow typically is about 10 times the weight of a human, we still use about 4 times more antimicrobials in livestock than in humans. And these are typically antimicrobials that are also used in humans. Some important considerations, as we need more surveillance so that we really know what is going on. Of course, infection prevention and control is very important. If people or animals are managed well, and people have hygienic circumstances, good food, then infections won't occur that often. And if you don't get this, let's say, viral infection, then you will get less secondary bacterial infection. So then there's less use of antimicrobials and also less occurrence of antimicrobial resistance. So we need stewardship, working together with all these parties that I showed. And also very important, we need research and innovation. We need new antimicrobials, we need new rapid tests, so that when somebody gets a sepsis, that we within 4 hours can know what drugs that we should use on those animals. So surveillance is really important. We need to develop the trends and identify then areas for intervention and understand the impacts. If you don't measure, you cannot just make targeted interventions. But it's complicated. This is a Canadian picture of the system that we do for monitoring. We monitor in humans, we monitor in animal populations, but you also see that environment and wildlife are not here. So we need better resistance, and we need to use the same tests to look at that resistance. And we fortunately can go now from the phenotypic tests also to the genotypic tests. We need both. So like I said, every infection prevented is one that needs no treatment, and every viral infection prevented is one that will not lead to a secondary bacterial infection. So if you give the right drug to the right patient at the right time for the right reason, that is antimicrobial stewardship. We need to go towards that. Quote of Scott Weiss and others. So just an example of how we do it here in Alberta. We work together with the Ministry of Health, the Chief Medical Officer, the Ministry of Agriculture and Forestry, so that is the Chief Veterinary Officer, and the Ministry of Environment and Parks, and together we work on antimicrobial stewardship. And it's amazing that also in the government you have those silos. The people that now are brought together have typically never seen each other, and they need to work together on this. An important last one, and BioGrid is in this sphere, is research and innovation. So we need to work together with research and innovation as well, that we get the new drugs and new diagnostics. That is really important. We have now these these bacteria that are not susceptible anymore for nearly any antibiotic. So we need to work on getting the right ones. And we need quick diagnostics. And we need to work together, like I said, because a boat with many holes and one patch will sink. So in summary, antibiotics used in different sectors, we should not think of that as separate actions. If we use antibiotics in animals, it will affect antimicrobial resistance in humans and in wildlife. So antimicrobial resistance in one sector has the potential to extend to other sectors. So we need to work together and work together all over the world. Like I said, bugs don't stop at borders. We have seen that with COVID-19. And I'm still ashamed of the COVID-19 vaccination efforts that were done in the very developed countries and then not done in the less developed countries. We really need to work together. Thank you.
Thank you. Say, please go ahead.
Yeah, thanks a lot, Professor Barkema. I think very insightful presentation. I'm sure there are quite a few questions that will come up, both from our side as well as from the audience. So we can quickly have Professor Urmi Bajpai to take on the next session. Thereafter, we will have a couple of questions for you and Dr. Urmi. Dr. Urmi, yeah, you can start.
Sure. Thank you for having me. And I think Dr. Herman has done half of my job, so I can quickly go through my slides because half of my slides are what he has already talked about. So I'll have to be careful that I don't present anything redundant. So give me a second. So can you see my slide, right? So thank you for having me here. My talk is about the management of AMR, and I am going to now mainly going to speak about bacteriophages because Professor Herman has already spoken most of what I had planned for today. But still to run through the first few slides. So as we saw that the COVID-19 has...
Dr. May, could you take it to the slideshow mode, please?
Oh yeah, sorry. So the antimicrobial resistance, the two things which Professor Herman has said is that it is not merely a medical problem, a health issues for humans, but it is also social and economic problem. And we saw that how COVID-19 further worsened the situation due to widespread overuse of antibiotics. So we are just coming out of the pandemic, and we have every reason to be worried about the misuse of antibiotics. And we see that if you look at the global map of AMR spread across different continents, India has a very a big, one of the most vulnerable countries amongst the other countries. And, we talk about discovery of new antibiotics, but, at the time when Alexander Fleming had discovered, he had this premonition that there'll be misuse. And he went on to say that a thoughtless person playing with penicillin treatment is morally responsible for the death of the man who finally succumbs to infection with the penicillin-resistant organism. So he kind of summarized and perhaps foresaw what was coming, that misuse can lead to the suffering of those who have never been exposed to. So the World Health Organization is the lead organization in managing and telling us the mandates. And let me see. So here, one is that, it was being said that AMR is one of the top global public health threats. And now I think it has come down to 3. It's one of the 3 global public health threats facing humanity. And it also lays a lot of emphasis on not just the misuse and overuse, but also the lack of clean water and sanitation, because that all leads to inadequate infection prevention and control, and which in turn leads to the emergence of resistant antimicrobial treatment. So this is one sector which also needs as much attention. And also that how the AMR has huge bearing on the economy. So here we see that, because it's like if we just focus on human infections and their suffering. So, in addition to the death and disability, there are prolonged illness, longer hospital stays, and expensive medicines, loss of working hours. All of these aggravate the situation, which is usually not, perhaps it's not visible to all. And also one more important thing is that we don't discuss antimicrobials only in the context of infectious diseases. If you see that if because of the emergence of resistance to antimicrobial to the antibiotics, there are pan-resistant strains, and in the wake of these resistant strains, even the surgeries, the major surgeries, the organ transplantation and cancer therapy, these all are inconceivable because they can catch infection also. So it's not just about the infectious disease, but also the non-communicable diseases, which will be impacted because of the antibiotic resistance. And that tells how grave the situation is. Now, how does AMR spread? So, that because the antimicrobials are given to food producing animals. And I'm not sure what kind of stewardship happens while prescribing or adding antibiotics in the feed of animals. And this is from where it enters the food chain and this cycle goes on. So as mentioned in the previous talk, that interconnectedness is very important. And so, for the infectious disease experts, the wake-up call came this year in the Lancet study, where they studied in 2019 the deaths because of antimicrobial resistance. So earlier, you'll see the data, death statistics for other diseases, but usually not a very systematic analysis has been done on deaths because of antimicrobial resistance. And they found that, the 6 microorganisms, bacteria, they alone were responsible for a certain number of deaths in America. And they found about 1.27 million deaths, which were attributed to AMR in one single year in the US. So one can imagine what would have been the magnitude of this deaths and the suffering because of antimicrobial resistance. So this Lancet study was sort of a wake-up call, and it drew a lot of attention to everybody that it's not about single deaths, but it is deaths because of single infections, but just because of the resistance. There are so many deaths occurring, and these statistics were alarming. So if you look, just this figure alone tells that the deaths because of antimicrobial resistance were far more than from malaria or from HIV and AIDS. And this was startling for everyone. So one more thing which we need to look at is when we look at these data is that we are seeing the impact of climate change, and it is in your face. The antibiotic resistance still is perhaps not visible to common people, but the climate change is very much visible. And we have to now also look at that how is this rapid change in climate going to impact the antimicrobial resistance menace? And here because there's an intersection between the health and particularly between the antibiotic resistant infections and the climate change. So just to give an idea that if the temperatures rise, so it's a very cyclic thing. So when the temperatures rise, and we all know it, so it can lead to floods, as we just saw in Pakistan and some parts of the world. It leads to droughts, it leads to wildfires, etc. And so you know that what happens after floods. So normally we don't study floods and droughts and AMR together in the same breath. But you know that if there are floods, so they'll cause the disruption of the sanitation infrastructure, which in turn can cause, also can cause huge population displacement and crowding, and also can result in waterborne infections. So imagine this vicious cycle that all of these will lead to the spread of antibiotic resistance. And we know that the dissemination of the sewage, especially in these conditions, which is the reservoir of antibiotic resistance genes. So all these calamities which are coming because of the climate change can also turn an ugly head for the AMR. And so also the population at risk for vector-borne diseases. So for example, those who have never been exposed to vector-borne diseases will get exposed because of this kind of environmental conditions, which in turn again can lead to antimicrobial infections, prescription of broad spectrum antibiotics, and resistance. So it is so intimately connected that I think, as Dr. Burman said, that all of the specialists have to come together to see how one condition is impacting the other. So what's the way forward? It is a grim situation. Sorry for a Friday evening to tell a very grim picture, but I think most of us are concerned, and that's why we're here. So the way forward is, of course, one is to work on the gaps, which include the expanding of the public health force and also the diagnostics, as has been emphasized enough. We need quick diagnostic tools, because in the absence of diagnostic tools, what happens is either they're very expensive or they're not available. Either way, what happens is antibiotics are more easily available in comparison and are cheaper also. So then there is overprescription and incorrect diagnosis and overprescription of broad spectrum antibiotics. So that is a matter of concern. And this I wanted to bring up, though this bill has not been passed, but to incentivize the discovery of new antimicrobials, we need some out of the box thinking. We need to see that how to make it an economically viable model. And for that, this interesting bill has been proposed in US, though it has not been passed yet. So this is called Pasteur Act bill after the French microbiologist Louis Pasteur. And this is about that this bill pays, it is about that it is it's a subscription model where the government pays for a certain amount and takes care of the expenses borne by the company, whoever, whether it is a biotech company or a pharmaceutical company. And this is based on public health value rather than the volume of antibiotics sold. Because very often, since we know that antibiotics are used only for a few days, so their sale is not as much as compared to the lifestyle disorders. And hence, they can never make up for the investment that they've done, and they're often going to bankruptcy, even if they come up with a good lead molecule. So if the government comes up with a proposal where there's a reimbursement reform, and that's where the expenses of the company is taken care of, then it can hugely incentivize startups and biotech companies to invest in finding new drugs. So this is, I think, a very thoughtful act. I hope it is not only passed in the US, but is also replicated in other countries. And of course, we have to populate the antibiotic and non-traditional pipelines. So having said that, this all is covered by already, so I will skip this slide. The systemic management also is important, where we have to look at the CDC and the WHO reports, and they have listed several methods of curbing the menace of AMR, which is like including telehealth for contact tracing. I think we can also pick up certain models which were followed in COVID-19 management, because several infectious diseases these are also bacterial infectious diseases. That was a viral infectious disease. So I just wanted to bring notice to interactive tools which are being really suggested by like CDC and Pew Charitable Trust. For example, Spark. So Spark is a cloud-based virtual laboratory that enables scientists to share insights. So as Professor Berkeman, Professor Berkeman, you have already said most of what I planned to say. So you were saying we are in silos, we need collaborations, interactions, and leveraging each other's strengths. So that is what exactly is required. And sometimes when you can't do physically, you can do in virtual mode. So this concept of developing cloud-based virtual laboratories where the scientists can share insights, and in fact, even the negative results and the antibiotics which did not work, that kind of data is not publicly available. And so that can save a lot of time. So these are some of the ways one can look at how to promote and expedite the discovery of new drugs. This slide is just to show you, this slide is a linker between my AMR and bacteriophages. So I don't know if you can see this. Wait, so here you see that this is a 2020 antibacterial agents in clinical and preclinical development. There are in all 43. Out of those, 27 are non-traditional. Amongst those non-traditional ones come the bacteriophages and lysins. So I will just briefly tell you what bacteriophages are, because my lab and my works on it, and many Indian researchers many researchers across the globe are working on bacteriophages now that antibiotics, we are running out of antibiotics. So these are viruses of bacteria. So they are predators of bacteria. And as they say, enemy of enemy is your friend. So a virus which kills a bacteria is good for you and is an antibiotic candidate. So if I'll try to run this slide, this is just to show that how effective this is. A Staphylococcus aureus infected with a fluorescent labeled virus is getting killed in a very short time, is being shown in this beautiful clip, just to give you an idea how bacteriophages kill bacteria. And they are nature's abundant, they're present in most numerous biological entities in nature. And these are very, very specific to antibiotics. And they were discovered in India. I mean, they were not known as bacteriophages then, but a British physician Ernst Hankin had found them first time in Ganga water in India. And he found that the bactericidal activity of water. So the bacteriophages, they use their bacterial host as a factory, make copies of themselves, and infect the new cells. And this is how they can clear a bacterial infection because of their biological nature. And the bacteria and the bacteriophage, they have co-evolved in the last few billion years. So they both are very used to of each other. They know each other well, they co-evolve well. And so it's a very dynamic interaction that they share. Now, if we compare antibiotics with phages, and phages being viruses and antibiotics being chemical entity, so we know that the bacteria gain resistance, which is what we're talking about, but bacteria can gain resistance against bacteriophages also. But given the abundance of bacteriophages in nature, you can discover the timeline for discovering new bacteriophages is very short in comparison to timeline for bacteria for antibiotics. Also, the investment required for developing antibiotic is far more than what you need for phages. And one of the biggest advantage of using bacteriophage is that they're very specific to their target. So the broad spectrum antibiotics versus a very specific phage. And that also makes sure that all the toxicity that comes along the antibiotics are spared in the cases of bacteriophages. And this event, I think I'll skip, it's a busy slide, but it tells the major events in the history of phages antibiotic discovery. And in fact, in the same time, around 100 years ago, a lot of new antibiotics were discovered by 1928 by Alexander Fleming. And around the same time, the bacteriophages were discovered also. So I don't know what was so special about these 2 decades. The first 2 decades, rather the second decade of 21st century, and now at 20th century. And I hope that something might be happening in present century also. We are in 2020s. This I'll skip. So basically this slide is a collage which tells that bacteriophage, those of you who have not heard of bacteriophages as potential antibiotics, so a lot is happening. It is happening across the world. Earlier it was confined to USSR and Eastern Bloc, but now it is in the Western countries also, and virtually every country now is taking up phages. And one can see investments happening, clinical trials supported by NIH happening, various biotech companies engaging in bacteriophages and derived products. These are the products from the yesteryears, those in the black and white. And these are the preparations which are made available by companies like Iliava Phage Therapy Center, which make the phages available for, I think, entire world, because they are the only ones who are amongst the few countries who are practicing phage therapy since last 100 years. Now, briefly to my lab. So we also work on mycobacteriophages. We develop bacteriophages bank so that they can be used potentially as a therapeutics in near or distant future. And for bacteriophages to be used as therapeutics, we need to characterize them in many ways. So here it is just shown that how should you characterize for them to qualify as therapeutics. And the license, now bacteriophages are the viruses, the whole entity, much bigger. And they also make proteins, which are called as lytic proteins. And these lytic proteins are enzymes called as lysins, which are capable of breaking down the cell wall of bacteria. So the way antibiotics can interfere with the cell wall synthesis or breakdown, likewise, the lysin enzymes can also damage the cell wall of bacteria. And if one damages the integrity of bacteria, then it is as good as an antibacterial. So antolysins and bacteriophages are the 2 things we are working on. Across the world also, we are finding several bacteriophages and lysins which are in different stages of clinical trials as listed here. So these are lysins and bacteriophages by companies, biotech companies, most of them. And these are the two of the most promising candidates. One was in the clinical trial phase 3, one in the phase 2. And there is a huge diversity in the landscape of lysins because they are found in nature in diverse, in abundance, and also huge diversity they show, which can be seen in their structure. So more diverse they are, more variety they offer, and hence they are likely to be more, you have them in pipeline for screening. And you can also study their antibacterial effect, as the figure here can show dramatically that if you treat cells with lysins in the center, you can see that there is no, in the presence of the treated cells do not show up at all, which tells the efficacy of the lysins also as good as antibiotics. So they have huge potential, and we need investment and collaboration to develop them as therapeutics as a substitute for antibiotics. So this is the IBRC roadmap for developing phages and lysins. So one is the collaborative interface is required between researchers and clinicians. And the steps in between require, building of banks of phages because they're found in abundance. So we can have large number of people engaged in building banks so that they can be made available as therapeutics as the need arises. These are the various lectures which are organized for dissemination of information about bacteriophage research happening around the world. And this is my team from my lab. So these all students below at the bottom on my left and right have been contributing to Phage Bank and to the license research. So I thank you all for your time. And these are like science art created by phage researchers again. So my last message is that prevention is preparedness. So if we prevent the antimicrobial misuse, we can prepare ourselves for a better future. Thank you very much.
Thanks a lot, Dr. Urmi. I'm sure there have been quite a few questions from the panel. Very insightful, very interesting. But before we hand over the questions to the audience, I would like to have a few questions myself, which have been put up by our team. The first question is to you, Professor Barkema. Would it be possible for you to elaborate a bit more on the One Health consortium that you're heading on the University of Calgary? What are the activities and what are the priorities out there?
So thank you for that question. So the AMR One Health Consortium is a consortium in the province of Alberta. We work all together with 4 different universities, Agriculture and Agri-Food Canada, and also with agricultural colleges on antimicrobial resistance. So we have a budget of about $17 million, and we have at this moment 36 different projects that go from discovery and innovation, new antimicrobials, new diagnostics, all kind of things, to actually clinical implementation in animals and in humans, to policy and also economics. So we have the whole gamut, and we have people from the whole spectrum also involved. And we work together on these projects. So all the projects have at least animal science, human scientists, or environmental scientists working together, but those could be basic scientists, could be clinicians, economists, policy specialists, all together. And that's funded mainly by the government, but we're now also getting projects in where the funding comes from the industry, that is pharmaceutical industry, but also the livestock industry. So we have about 105 researchers at this moment, more than 100 trainees, so that's graduate students, postdocs, and then in the summer we have a lot of summer students also, and a huge amount. And so, but it's really the working together that I really like, where we also have people working on bacteriophages. That's a cool area, and I really like the talk of you, Urmi, by the way.
Thanks for that, Professor. I think very interesting work being done at your end. Dr. Urmi, now's the era of personalized medicine. And so what do you have to talk about personalized phages as a concept? You need to unmute.
I think the personalized phages, because they are so specific, they will mostly be used as personalized medicine only.
Okay.
Yeah. In fact, we have a good model in Belgium, the magistral preparation, which is now available in pharmacies. And once the doctors prescribe, the pharmacist can give it to the patients. So personalized medicine is the way to go. I think in coming time, when the aging world population and chronic infections, I think we don't have any other way.
Dr. Barkema, my question to you would be, when it comes to pathogens affecting human lives, WHO is very clear. They have drawn up a list of high priority pathogens, those which are critical in terms of AMR. We also talk of escape pathogens. How is it in the animal world? Does FAO and OIE, have they drawn up a list, or is it very similar to what's happening in the human world?
It is very similar, but with different pathogens playing a role sometimes in different species. Of course, the focus right now, and so if human health organizations like WHO think of a One Health approach, they particularly think of prevention of infection in humans from animals, while veterinarians like myself also think of prevention of infections in animals because of the health of those animals. and also because of the economics of the agricultural sector. So we look at both, but so where I focus mainly on is prevention of infection. And we can really say, okay, we cannot use preventive use of antimicrobials anymore, but then the management of these herds need to be such that you don't need that umbrella of antibiotics anymore, because we also need protein for the people. So we keep the livestock for a reason, and that is to feed humans. And so we need to keep that in mind. So I think we have a duty as veterinarians, agricultural scientists, and others to work with people all over the world, helping them to increase management so that we don't need that preventive use of antimicrobials anymore. And can I just put a plug in for the seminar series that we do from the AMR One Health Consortium? Some of you that are present, I recognized your names, were participant in these seminars. So we have a seminar series that is by donation only, so people that don't have a lot of money can still participate. It's 24 seminars on AMR with a One Health approach. And if you go to the website of the AMR One Health Consortium, or you go to LinkedIn, you will find us. And we have experts from all over the world presenting there. And this year we have more than 200 participants from 52 countries. So and we actually made it the time so that people also from India could attend without losing sleep. So
Great, thanks. I think we would like to disseminate this information about the series that you were doing for the benefit of the Indian community out here. I think there are a lot of requests from the participants on whether the slides would be shared. We would not be able to comment on that right now. We will have a discussion with the panelists and IBLoT. And if not the whole presentation, at least part of the presentation which the panelists feel is not confidential will definitely be shared. So you can write to us for that. Professor Urmi, one question out here from the audience is whether phages do infect the non-pathogenic bacteria in the body.
So phages will be administered only for the infection, which is for the bacteria which is causing the infection. So of course we have inside our body phages and bacteria both. In fact, phages help us they modulate the microbial environment, and they also maintain the microflora. They prevent the opportunistic infection. So that is their normal role. But when the phages are administered as therapeutics for therapy, they are targeted against the infection causing bacteria. So the procedure is like that. First, the way we do antibiograms, likewise, we test the sensitivity of the clinical isolate, sensitivity of phages against the clinical isolate, and those phages are administered. So we don't fear them infecting the non-pathogenic bacteria because they're not targeted.
So these are very targeted, very specific.
Very specific.
Yeah. Just one more question for you. Is it possible to engineer lysine CDSs of different phages in a single phage genome to use as a master phage instead of using a phage cocktail?
Engineer lysine series of different phages in a single phage genome to use as a master phage instead of using phage cocktail. So, first, the reason why we use phage cocktail is because if we use a monophage therapy, then the probability of bacteria, the target bacteria gaining resistance to phage is very much there because, they are co-evolving all the time. Sometimes monophage therapy is also successful. It's not that they're not, but there is a probability. So we use phage cocktails to ensure that, if you send in 3-4 phages targeting the same bacteria, then the probability of bacteria gaining resistance against all 4 of them is remote. And that's the reason why I use phage cocktail. Now, if we put in the lysine genes, if I understand the question correctly, if we put several, let's say, very effective lysines within the one phage genome, but then the resistance is happening outside. Like, for example, if the bacterial mechanism of resistance to phage is that it just shuts off the receptor, it doesn't bind to the phage. So what will we do with those extra lysines inside the phage? So bacteria can confer resistance to itself against the phages using several mechanisms. We don't know which one it is going to use. So I think what the person is trying to say is make the phage a superphage, but then the superphage can be rendered ineffective in very short time because bacteria may not be happy seeing its receptor binding protein. So if it does not bind to the phage, however effective the phage is, it will be useless. So yes, we do engineer phages to increase their lytic ability, but that will not compensate for cocktails. Yeah, yeah. Engineered phage is very welcome as a better therapeutics, more efficient, but it's replacing a cocktail, that is unlikely.
Yeah. Thanks, Dr. Professor Herman has to leave because it was a seminar scheduled for an hour and we are well past an hour. Doctor, if you don't mind, just one last question. This is a combination of 3 or 4 questions that have been directed to you to save time. I've just clubbed them all. We're saying, well, so many stakeholders are involved. You have the FAO, you have the OIE, you have WHO. And in each of these countries, we have animal health being taken care of by the Ministry of Agricultural Sciences, human health being taken care of by the Ministry of Health. So how do you approach the concept of One Health when so many different stakeholders are involved? How would you go about a policy advocacy or a protocol, regulations, guidelines? Isn't it a daunting task?
It is a daunting task, but that doesn't mean that we should not take this task on. It is really worth taking this task on. And it all starts with having objectives, clear, measurable targets that we go towards. Like the UK did with, okay, we are going to decrease the use of antimicrobials with 25% in 5 years, or the Netherlands, my home country where I came from, with 50%. And if you then make those targets and you agree with all the organizations on it, then you have to work together to make it happen. Yeah, just sitting together and talk of, okay, what are we going to do? Yeah, doesn't really lead to progress in that area. You need to have targets. And we in Canada, we are forming here this organization also to work on it. And we identified 1,200 different partners that we are working on, because every province has their own dairy organization, veterinary organization, dentist organization, pharmacist organization. So name it. Yeah, but so if you together all have this target, then you get somewhere. So we need a focus.
Yeah, there are a lot more questions, Professor Barkema and Professor Umi. But what we would do is we would request the audience to write into IBLoT or also write into connect@techinvention.biz. One of my colleagues will put that up on the chat box. So we will collect those answers and have it mailed back to you. I would now like to hand over the floor to Yogesh. It has been a wonderful having the two of you from different perspectives, and I will have to disappoint the audience a bit because there are so many questions that have come in directly, even outside of the common chat box. But we will promise to bring you more seminars, more webinars, more insights as we go about. Yogesh, over to you.
Yes, I'll be taking over for Yogesh. He's caught up a little, but thank you everyone for attending the seminar today. I thank you, Syed, for moderating this. Thank you, Dr. Barkema and Dr. Umi Bajpai, for being our guests for honor today and for disseminating such useful information. Again, to all the participants, you can definitely shoot out your questions over LinkedIn, Slack, etc. Right, and we'll make sure we send these questions to the right representatives as well. And thank you, Techinvention, and all the other collaborators we've had today for hosting this event and collaborating with us as we look forward to disseminate information to help grow ecosystems, not just in India, but across the world. Right, and so with that, I'd like to end this event. We have an upcoming event from IBLoT along the lines of AMR with Dr. Anand Kumar from Bugworks. And I've posted the link for that event as well in the chat, along with our LinkedIn and Slack channels. And Syed and Dr. Barkema and Dr. Umi, if you have any links or events that you want the participants to look forward to, you can either post it on the chat or share it with us, and we'll share it with the greater audience. But with that, thank you everyone for participating. Thank you for all the responses. And of course, please keep track of your questions and send it to us. We will definitely reach out to the speakers and the moderators to get it answered.